Metastatic pancreatic cancer is one of the fastest-moving diseases on the planet. By the time it reaches a secondary organ, like the liver, the tumor cells are multiplying and overwhelming surrounding tissue at a rate that standard treatments struggle to match. When Jan Bizian arrived at the ER with persistent stomach pains, he received exactly that diagnosis. Statistically, roughly 2% of patients in his condition live past 5 years. His doctors started him on FOLFIRINOX, a grueling multi-drug chemotherapy regimen. Halfway through the treatment, medical scans showed it wasn't working at all. The cancer had continued its advance into his liver. Then, his body responded. The tumors retreated. Today, 9 years after a terminal diagnosis, Jan is alive and cancer-free. Modern oncology spends billions analyzing how and why cancer kills. But because cases like Jan's are extremely rare, medical science has historically lacked the data to investigate the specific biology of those who live. A biotech startup named Cure51 is targeting that exact blind spot. Instead of mapping the progression of fatal tumors, they are actively hunting down and studying cancer super survivors. They call this effort the Rosalind Study, honoring the scientist Rosalind Franklin, whose work provided the first clear evidence of DNA's helical structure. The project is actively gathering a multimodal database of over 500 clinical outliers across more than 50 medical centers worldwide. Researchers take the biological profiles of these exceptional survivors and match them directly against a control group, patients who experienced standard fatal outcomes. By running this direct comparison, the Rosalind Study acts as a forensic investigation engineered to isolate the specific biological advantages that saved these outliers. To find these survival mechanisms, researchers use Multiomics to map millions of data points across 4 distinct tumor layers. First is genomics, reading the cancer's DNA code. Above it, transcriptomics maps exactly how that code is expressed. Then comes proteomics, the protein machines, and microbiomics, identifying the bacteria acting as friends or foes inside the tumor. This technical readout shows the results of that mapping process. By feeding these layered models into artificial intelligence, computational biologists use graph-based clustering to categorize cellular differences that would be invisible to the human eye. Multiomics allows researchers to stop guessing. They can actually see the precise biological mechanisms that make an outlier's tumor vulnerable. This computational approach is already pointing toward concrete biological anomalies in long-term survivors. Looking at this cancer cell diagram, the left side illustrates the yellow STAT3 protein, which typically promotes tumor growth and shields it from the immune system. But in outliers, their bodies suppress STAT3. As shown in the middle and right sections, a blocker substance inhibits the protein, disabling the shield and forcing the cancer to destroy itself. Finding suppressed STAT3 identifies a potential vulnerability that drug developers can now focus on. The goal is to eventually replicate this natural resistance in patients who lack it. The final step of the Rosalind mission is translating these outlier anomalies into scalable drugs. If researchers can map the protein blockers or genetic overrides that naturally occur in a super survivor, they can develop treatments to artificially induce those same states in standard patients. This reverse engineering process is now being applied across multiple lethal diseases, including glioblastoma and extensive-stage small cell lung cancer. With a growing data set of over 1,200 identified outliers, scientists are building a map of the biological exceptions to terminal disease. By decoding the secrets of super survivors, science is attempting to transform biological luck into scalable medicine, pursuing a future where survival is no longer a statistical anomaly.